My name is Darla Martinez, and I am investigating the influence of cannabidiol, CBD, on gene expression in human peripheral blood mononuclear cells, PBMCs. The focus of this research is to test the hypothesis that CBD alters the expression of genes that are important to the functions of PBMCs. Previous work in the Kamensky lab identified genes that were sensitive to modulation when exposed to various CBD concentrations. These identified genes were BTN2A2, BTNL9, and PPL. To test this hypothesis, PBMCs were isolated from blood donations. They were then suspended in complete RPMI media. Cells were treated with DMSO, the vehicle control for CBD, and CBD at a concentration of 32 micromolar. A naive group, which had no treatment applied to it, was included for each donor and served as a control for the vehicle. Cells were incubated with the treatments for 6 or 24 hours. After the incubation period, cells were harvested and lysed with a lysis buffer from Chiagen RNA-C plus minikit. Once cells were lysed, RNA was isolated using the same Chiagen reagents. After RNA isolation, the RNA concentration for each was determined to then proceed with a reverse transcription reaction, which converted RNA to cDNA. After cDNA was isolated, it was diluted 100-fold, and this diluted cDNA was used for quantitative PCR to determine gene expression using primer and probes for genes BTN2A2, BTNL9, and PPL. The data presented in Figure 3 illustrates that the 24-hour incubation time point proved that the treatment of CBD was inconclusive. Also, the data was statistically insignificant, which was determined by a one-way ANOVA test. The 6-hour time point, which is shown in Figure 2 and 3, showed a decrease in BTN2A2 mRNA that was expressed after treatment with CBD. This data was statistically significant, and this was also determined by a one-way ANOVA. Also, in Figures 4 and 5, these are amplification plots from the quantitative PCR. These figures illustrate the lack of PPL and BTNL9 expression. We know that these genes are not expressed by comparing the curves of the 18S, which is always present in all organisms as it is a housekeeping gene, as well as the expression of BTN2A2. Based on the data collected, it can be concluded that CBD decreased mRNA levels of the gene BTN2A2 at a 6-hour incubation period as opposed to a 24-hour period. Also, it could be concluded that previous genes that were shown to be modulated by liver cells, such as BTNL9 and PPL, were not expressed in human PBMCs. More samples and more testing needs to be done to concretely confirm this initial finding. This research is ongoing, and future work is focusing on culturing Hep G2 hepatocytes and to a level of maturity to allow them to be subjected to similar treatment as PBMCs to determine if similar patterns of expression will be noted between liver hepatocytes and human PBMCs.